Hello. Right, so the next practical that you ought to have done if you've been able to come into college concerns growth media. The growth media, as we've said, just means food for bacteria, but it comes in different sorts and for different purposes. There's a PowerPoint on the different types of media and their different uses on the Google Classroom. Now, growth media generally come pre-prepared in powdered form, and there are different types of media for different uses. So in the practical that you would have done, we would have worked with 4 different growth media and 2 different bacteria to identify the best growth medium for both of the bacteria. Now, we have 4 types of growth media that we would have tested. 2 of them are what we call complex media, which basically means they're made from living material. We don't know exactly what chemicals are in them. One of them is what we call a defined medium. This one is called Xapex. That means it is composed entirely of chemicals from a store cupboard. We know exactly what's in it. In this case, for example, this is made of sodium nitrate, potassium chloride, magnesium glycerophosphate, ferrous sulfate, potassium sulfate and sucrose, aka sugar. So we know exactly what's in this one. The other 2 that are standardly used are nutrient broth and peptone. Those are made from meat, and so we don't literally know every single chemical that's in them. They're basically soup. And the final one that we are going to investigate is what we call a selective medium. It only lets certain bacteria grow. It is a type of medium called McConkey. And what it is supposedly only allows gram-negative gut bacteria to grow because it contains bile salts, which are toxic to a lot of bacteria. It also contains a type of dye called bromocresol purple, which kills most gram-positive bacteria. As well as that, it's got the basics of nutrient broth. But the important thing is the bile salts, which kill most bacteria that don't naturally live in the gut, and this dye, whose name I can never remember, bromocresol purple, says so right here, which will kill most gram-positive bacteria. So the first thing I would have got you to do, because it's like a 3-week practical, is to make up the solutions. You would weigh out a certain amount of the powder, dissolve it in water, put it in a bottle, such as this, a heat-proof bottle, and then the next step would be to sterilise your liquid by putting it in an autoclave. Now that's been done for us, if you like. So we have here the finished results, the made-up growth media, the 4 different possible things we could grow the bacteria on. So we have nutrient broth, which is the standard food for bacteria. It's basically beef extract, yeast extract, some sugar, so soup. The point is it's got vitamins, there's also minerals added, salts and stuff, calcium salts, all sorts of stuff they need. Anyway, it's supposed to be super duper food for bacteria. The next one is peptone, and that is basically beef stock. You know how in Victorian cookbooks they say, make beef stock, get some beef, boil it for 18 hours. The reason they say boil it for 18 hours is the proteins start to break down into short, what we call peptides. And that's what this is. It's beef that's been boiled so long the proteins are starting to fall apart. It's not even gelatin anymore, it's just sort of little bits of a little short lengths of amino acid. Then we have the Zapex Docs, which I've told you about. This is a defined medium. It's made of chemicals from the store cupboard. So yes, it's got sugar, yes, it's got minerals like magnesium and sodium that bacteria would need, but it hasn't got any proteins, any vitamins, any amino acids, any organic chemicals at all except sucrose, which means the bacteria have to make all of those for themselves. And then, as I say, we have this selective medium, which is supposed not to grow all types of bacteria. I say suppose because bacteria don't always read the textbooks when we try these practicals, but that's the theory. Right, okay. So, what would you have done had you been in college? Well, the first thing that you would have needed to do was to inoculate the different growth media with a culture of bacteria. So, I will demonstrate that. I've already cleaned my bench down, washed my hands, etc., and done all the normal safety precautions. So, come on, the gas is on. Yep. Right. So, what we are going to do now is inoculate the culture. Obviously, the idea is to place the same volume of the bacterial culture into the same volume of each of these growth media to see which one is going to grow the most bacteria. I'm not going to waste your time inoculating all four, but I will just demonstrate inoculating one of them. So we have here some sterile graduated pipettes. They're sterile till they come out the pack. And what I would normally get you to do is put 0.1 millilitres of bacterial culture into each of these. So obviously you would need to label it with your name, the date, bacterium, so that we know, as we've discussed, who is responsible for culture. So what I will do now is take out my sterile pipette using the correct technique that we've talked about. Flame the bottle, take up my 0.1 milliliter of bacterial culture, and it's not what I labelled, that's what I labelled. Add it using correct sterile technique to my bottle. Okay, obviously pipette and disinfectant. And I would obviously repeat that. I would have got you each to repeat this with the 4 different growth media. What happens then is the 4 different growth media, all inoculated with the same amount of the bacterium, would be placed in an incubator, typically for 48 hours, maybe a bit more, and then refrigerated until the next week when you would have come to do the next part of the lesson. Right, so we will assume that has happened. Okay, but I'm not coming back next week. Right, so we will assume it is a week later. Here is the bottle that I just inoculated, which has been incubated and is now grown a fair number of bacteria. Now, what we have to do now is count how many bacteria. There are 4 different methods that are sometimes used, and I'm sure there are more somewhere in a textbook, but there are 4 standard methods for finding out how many bacteria per milliliter there are in this bottle, so that we can compare it to the number of bacteria that have grown in the other growth media. The method I'm going to use today is called viable cell count. The other methods we will discuss, and there's going to be a separate video on those, which you'll find on Google Classroom. But basically what we are going to do here is we are going to take this culture and we are going to dilute it 1 million times in sterile water. When we've done that, we're going to spread that 1 million times dilution on an agar plate. And because we've diluted it 1 million times, it'll just be a few dozen maybe bacteria. So it'll be like one there, one bacteria there, one there, one there. Then we incubate the plate. And when we come back next week, where there was one bacterium on the plate this week, there's going to be a whacking great blob. But blob isn't a science word. What science word is colony. But you get the idea. There's blobs we can count. And I do have one over there that I prepared earlier. I will show you at the end of this video. Right, so how do we do this 1 million times dilution? Well, the first thing we need to do is get some test tubes which have been sterilised in an autoclave. Get a bottle of water that has also been sterilised in an autoclave, and put 9.9 millilitres of water into each of our test tubes. Obviously, again, we need to make an effort to keep things sterile, otherwise we shall get who knows what horrid bacteria rather than the ones we're after. But I can touch this end, because this end isn't going anywhere near my bacteria. In you go. And again, as far as possible, using correct sterile technique, a little harder with these large bottles. We will draw up 9.9 millilitres of water. There we go. And put it in our sterile tube. Like so. Like so. Not the way it wants. Right. This isn't particularly contaminated, but I'll just put it there for now. Right, so what I've got now then is a whole row of test tubes. Each has 9.9 millilitres of water. For each bacterial culture that you want to count the number of bacteria, you need 3 test tubes. And what I'm going to do is as follows. I'm going to transfer 0.1 millilitres of the bacterial culture that I want to count the bacteria of into the 9.9 millilitres of water. So I'll have 10 millilitres in here, and I will have diluted the bacteria 100 times. Then I'm going to take 0.1 millilitres out of this tube and put it in this tube. This one is behind the pipette, isn't it? It would be. Right, in this tube. Yeah. And then I'm going to take 0.1 out of there and put it in here. So I'll have 100 times dilution, a 10,000 times dilution, and 1 million times dilution. So it's a bit tedious to watch, but nowhere near as tedious as it is to do when you have to do this as we used to in the lab, when you had to do 12 or something of them because you had 4 bacteria. So let's rock and roll. Again, correct sterile technique. We will get 0.1 mils. 0.1, yes, mils of our bacterial culture, and put them in the first of our labelled tubes. Give the tube a shake to distribute the bacteria evenly. And then, as they say, lather, rinse, repeat. So I will now just carry on diluting twice more. The wrong end of the set. Yes, okay. The perils of wearing glasses and safety specs. Let's try that again, this time with the right end of the set. That's better. Yes, you know how you can't see anything with goggles over specs. Right, Okay, so again, correct sterile technique, 0.1 from this tube into this tube. So this tube, I've diluted my original bacterial culture 10,000 times. And I'll repeat that, obviously with a clean pipette, which I'll get the right way up. I think that's why one of the reasons they put cotton wool in them. I'm sure there's a better reason, but it doesn't help me to work out which is the right end. Right, so we will take 0.1 mils from the 10,000 dilution and transfer it to another tube. And in this tube we've diluted our original bacterial culture by 1,000,000 times. Great. So what we need to do now, clear away some of these things that I need no longer. Bear with me a mo. Okay, what I need to do now is my 10,000 and 1,000,000 dilutions. I want to plate 0.1 millilitres of them out onto an agar plate and spread it using the same method that we learned when we were learning about correct sterile technique a couple of weeks ago. So I won't bother doing the 100 dilution because usually there are so many bacteria on that if you spread them on a plate you just get the complete goo everywhere, solid bacteria. So I'm only going to plate out the 10,000 and the 1,000,000. So I've labelled my plates. This is the 10,000 working close to the Bunsen, remember? Is that that end? Right. Okay. So my 10,000 culture, I will plate that. $10,000 dilution, 0.1 millilitres, 10,000 plate. And spread it with a sterile spreader. I know I'm slightly behind the Bunsen burner here, but I've demonstrated this method already, so you're familiar with the idea of spreading a plate. Make sure I get in all the corners. Okay, I will sellotape that up. That's the best place to put the sellotape so that it wasn't in the way of the camera. Hey ho, bear with me. I will sellotape that up, two sides. I'll probably come back and do that better when I've finished recording. And then carry on and do my 1,000,000 dilution in the same way. Isn't microbiology boring? I believe we've been at this nearly 20 minutes. Right, so I will now plate my 1,000,000 dilution onto the other plate. Again, 0.1 mils of my 1,000,000 dilution on here. Spread it with a sterile spreader. And you'll be pleased to know that's pretty much the end of the method. So I'll tape that in a moment. Seen me putting tape on things before. Right, okay, there only remains then to go over there and get the ones I prepared earlier to show you the sort of results you might get. Wait there one sec. here are the ones I prepared earlier. And actually I really need something dark coloured to hold these up against. I should have thought of that, shouldn't I? We've got some black card. I'll go and get some black card. Right there. Black card. So, still doesn't show brilliantly. This is my million dilution, and as I hope you can kind of see, there's nothing growing on it. So obviously there wasn't even one bacterium in my million dilution that I prepared earlier this week. The 10,000 though, and again I'm afraid it's quite difficult to see, but if you have a good imagination, it's speckled with hundreds of tiny colonies. So what I need to do now is count how many colonies. And the best way to do that is over the black card, jelly side up. I sit here and I go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Putting a dot with a marker pen on each colony, over each colony as I count it, so I don't count any twice. And I'm not going to make you watch me do that because I reckon there's at least 1,000 colonies on here. However, if I count this and I find for 1,007 colonies, that tells me that on Monday there were 1,007 individual bacteria in my 10,000 dilution. If I then multiply that number by 10,000, that will tell me how many bacteria there were in 0.1 millilitres of my original culture. And had you been in college, I would have got you all to do this for 4 different types of growth media and 2 different bacteria. But I'm not going to do all of that now. I think you get the idea. I'll leave it to you to make a prediction, and I'll ask you in a minute when you've all finished watching this video what you predict I would have seen. Which of the 4 growth media would have grown most bacteria and why? Okay, right, so that is the end of this, which is probably the longest of our practicals. When you finished watching this, there are some questions on the Google Classroom, I hope. Right, thank you very much. I'll see you again in a while.